Hello, everyone, and welcome to today's webinar, Introduction to AI Image Analysis of Microscopy. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit them at leica-microsystems.com. Now, I want to encourage you to participate today by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain those credits. I'd now like to welcome our presenter, Dr. Quyen Tran, an Applications and Marketing Manager at Avia. Dr. Tran, welcome. You may now begin your presentation.
Hello, everybody. Welcome to the webinar, and thank you very much for joining us today. My name is Quyen Tran, and today I'll be doing an introduction to AI image analysis and microscopy. A quick look at our agenda. We're going to go through some AI basics and then specifically machine learning and how it can be applied in the microscopy field. We'll then talk about deep learning and how that is applicable, and then do a Q&A session so that you have plenty of time to ask me questions about the topics that I covered here and potentially how these tools and applications can be utilized in your particular research. So some overall terminology to kind of lay the foundation for us here. So artificial intelligence, or AI as you probably have heard it, is software that mimics the way we humans think and learn. Machine learning is a subcategory of this that is specifying algorithms that don't explicitly, that hasn't been explicitly programmed to complete a particular task. They, however, learn to do this task from training data. I hope that's going to be a little bit clearer as we talk more about machine learning and I showcase some examples for you. Now, another smaller and even more subgroup of this is deep learning. And deep learning really expands and allows the software to learn from a large and vast amount of images and data and information to complete the task at hand. A quick look at how traditional versus AI-based image analysis workflow is done. So in traditional image analysis workflow, we humans create the rules and feed these rules and the data into the program. So some examples of rules that could be created is that a cell has to be a certain level of intensity and a certain size to be counted. And we feed these rules and the data into a program, and the program provides us with answers. So segmentation and detection of the cells that we wanted. Now, with machine learning, we're basically flipping that around, and what we're actually putting into the framework is answers and data. So answers could be examples of what a cell looks like, examples of what a protein looks like, and we're feeding the examples and the data into the network and the framework here. And what it provides us are the rules that govern and characterize why the cell is unique, why it's different from the background, why it's different from the other cells or the other protein that you could look for that are in your image, essentially. So it's a collection of rules that really help us characterize our unique objects that we're trying to detect. So let's dive into machine learning and how it can be used in microscopy, and particularly talking about the random forest algorithm. This algorithm is one of the most commonly used algorithms when it comes to machine learning in microscopy. You probably have already seen it used in as a way to classify pixels or objects into different classes and different groups. And essentially, it's composed of multiple decision trees here that collectively allows the software and allows you to select or classify each pixel or each object. Now let's dive into what is a decision tree. It's a type of supervised learning, and it's essentially created and composed of a number of features that are critical to making a decision or a classification. So the example I have here is somebody coming in to apply for a loan, and some of the key features and key metrics that the person who's the loan officer may look at is the age you are, your education level, whether or not you're a homeowner, and the amount of income you bring in. And these features help the loan officer essentially decide whether to refuse this loan or to provide you the credit. So this is one decision tree composed of these set of features to provide a way to classify this particular application. Now, in the learning process itself, each of these features essentially gets a vote, and the majority of the voting of these features determines the classification and the output of this particular tree. And what we're really trying to accomplish with the decision trees is to group and create a set of features that allows us to best categorize and best split the data. So we're looking and trying to determine a set of features that really allow us to classify whether this is a cell or not a cell. And so how do we get from a decision tree to a random forest? And essentially the overall workflow is we take the entire data set with the labels, the examples of what a cell looks like, and then we subdivide this data into random groups and random subsets of the data. We utilize this to teach a tree, and each of these trees also have a random selection of features. So this tree is going to have a unique set of data points and unique set of features to allow us to make that classification of a cell or not a cell. And then essentially we repeat this process to create our random forest. And the goal here is that because it's a random selection of data points and features that make each of these trees are going to be slightly unique from one another. It's going to be looking at slightly different things with slightly different data, and collectively this decision tree and this random forest is going to allow us to come to the right answer of whether or not this is a cell versus a background or a different class of cell. So you can see here where the name and the terminology comes from, right? You're creating a forest of decision trees that are kind of randomly created, hence the name, the random forest algorithm. That's kind of my easy way to think about this and an easy way to remember this algorithm. Now, how does this get applied in person? We really just talked about kind of the theory behind it, but how do you actually use it in microscopy data set? So this is an example of what machine learning pixel classifier training could look like. So here we're creating a model that has two classes, what we want and the background. And we're providing examples of each of these two classes by essentially painting the pixels. Which pixels really characterize what it is that we want to detect, and then which pixels characterize that background class. So again, we're painting examples of these two classes during our training process. And then we create a random forest algorithm that allows us to basically classify each of these pixels for one of the classes. So we basically get an output once we create the model and apply the model that classifies pixel by pixel, is it background or is it what we wanted? And you can see with just a small set of example pixels that we provide to the random forest algorithm, we can basically classify the rest of the image into these two classes. And it does a relatively good job about picking up what it is that we want and what it is that we classify as background. Now this example only has 2 classes. However, you can make additional classes with the pixel classifier. Now, a few more examples of what image enhancement using the machine learning algorithm could look like. So this left example here is extracting multiple structures from a single channel. So here we have a protein or a marker that essentially has stained both the nucleus and the cell membrane in relatively the same level of intensity. So if we were to apply a intensity-based algorithm here, it would be really difficult to kind of divide and separate these 2 components. However, as human beings, as we look at this, we can extract the other morphological characteristics from it, right? The nucleus is more globular, circular, where the cell membrane is more straight and thin. And so essentially we can use the pixel classifier to take advantage of some of those morphological characteristics that I just kind of described in words, and paint examples of what the nucleus looks like, what the cell cytoplasm looks like, and then what the cell membrane looks like, and divide and go from essentially 1 single channel to three channels. The blue, purple for the cell nucleus, the red for the cell cytoplasm, and the yellow for the cell membrane. So now we have this fluorescent-like image that separates these 3 components that we can now further do subsequent analysis, such as cell counting, detection, things like that. We also have the ability with the machine learning algorithm to go from a heterogeneous staining to a more homogeneous staining. So in this particular example, and you can see a little bit more in the larger nucleus that this nuclear staining is heterogeneous and has a kind of more puncta appearance where there are areas of bright intensity and areas of low intensity. And this again would be really difficult to capture the whole nucleus if we were just using an intensity-based algorithm. So what we did here is we provided examples of what the entire nucleus looks like, and the pixel classifier kind of expanded this and allowed us to create a new channel that has a more homogeneous staining and evens out the staining and captures the full scope of the nucleus, so that subsequent image analysis protocols that we have that are intensity-based can now do a much better job at this detection. And then the example we saw with the training is that we have the ability to enhance certain objects of interest from the image that you have created. We can also go from a phase to a more fluorescent-like image. As some of you may have tried to kind of do this in your phase contrast image intensity-based algorithms, again, here are really difficult because there's not as much contrast between the cell and the background in terms of intensity. But if we use the pixel classifier, we can use some of the more morphological characteristics and pull that out and really create this fluorescent-like image that has a high intensity level for the cell and a low intensity level for the background. So that wide range and that variation really allows us to now do detection and analysis with intensity-based algorithms later on, and it makes that process a lot easier. There are also ways and tools that can allow you to go from the pixel classifier, using the pixel classifier to go from the image all the way to the segmentation, not just in the enhancement, but all the way to segmentation. And the examples I provide here are one, segmenting cells, in particular for this example, the nucleus in a histology image. And also the ability to segment cells in phase contrast image to perhaps help you do cell counting a little quicker and a little bit easier. And to also segment cells in fluorescent-based images. And I really like using machine learning for cell detection of cells that are kind of elongated or have a very unique structure to them. A lot of the algorithms that are traditionally used, that are commonly used, are optimized for more kind of globular cells, which makes it a little bit tricky if your cells don't kind of conform to that morphology. So the pixel classifier kind of allows you to expand that a little bit more and capture cell morphology that are elongated or just kind of irregular. I also really enjoy using the pixel classifier and the machine learning algorithms in general to segment and to detect cells that are kind of really difficult to partition based on intensity levels. And the machine learning algorithm just again allows me to take that step further so that I can separate these cells more based on more than just intensity and cell size. It allows me to add a lot more complexity to that so that partitioning is significantly better. So some key advantage that I hope you've seen from the examples I provided here, machine learning really allows you to leverage your biological expertise here. You are the one that's kind of providing the expertise and providing the examples of the objects that you don't want and what makes kind of the class that you want. And then because you're providing these examples through a painting method, it cuts down and reduces the knowledge of computer science that's required in terms of image analysis and in terms of using machine learning. It allows you to kind of, again, utilize your biological knowledge to provide those examples instead of those rules that may be required and may utilize discrete measurements that you have to figure out. And it allows you to kind of go beyond those discrete measurements, right, to characterize your objects and go from difficult images to analyze and making them easier to analyze. So even if you do have to use an intensity-based algorithm later on, you can make those images a little bit easier for the algorithms to process and to analyze. So you can, kind of have the best of both worlds, pixel classifier and machine learning to make it a little bit easier, and then the intensity-based algorithms that you're more familiar with and used to and have developed a pipeline to process the rest of the images the rest of the way. So now let's kind of shift gears a little bit and talk about deep learning and microscopy, particularly the convolution neural network. And this neural network is really commonly used in AI and deep learning in relationship to the microscopy field. And essentially the structure of it is it's composed of input layers and hidden layers, and these hidden layers can range in numbers quite a bit, and then an output layer. And I hope that you'll be able to see a little bit when I show you the next video is that this network really kind of resembles and mimics the way neurons work within our brain, especially how they signal to one another. So this video is gonna walk you through some of the basics of what a convolution neural network is and also introduce you to some key terminology that you may have seen in this literature. So what I showed you is just a brief glimpse of this field of research, and researchers continue to push the boundaries of what deep learning can do for microscopy data sets by developing new types of architectures. I'm just providing some examples you may have seen before: UNet, ARCAND, CARE, to kind of utilize deep learning for a number of applications in microscopy. There are also a number of researchers who are looking into different types of networks to, again, push that boundary of deep learning and microscopy. So I won't be able to cover everything here, so I really encourage you to kind of explore this a little bit more. There's a lot of really groundbreaking and exciting work that's happening in this field that is really taking the next steps between what we can do in terms of microscopy, in terms of visualization and analysis, and really taking it to the next level. So I highly encourage you to look into this a little bit more. Now let's talk about and let's kind of see deep learning in action. So let's go back to kind of what deep learning could look like and usually looks like. So it usually starts out with what we call an input image pair. This particular example is a low signal to noise image and a high signal to noise image as this pair. And it essentially gets fed into a neural network. And this neural network can be different depending on what it is that you're looking for to do and what works kind of best for your data. And essentially all this outputs is a deep learning train model. So it's that set and collection of rules that we talked about earlier. And this allows you to now add in more data and apply this model to create, in this particular case, a restored image. So this workflow in this model is really created to take a low signal to noise image and turn it into a high signal to noise image. So that's why when you apply the model here, you get this restored image. I also want to briefly show you what this could look like for EM segmentation. Just because the image pair here is going to be slightly different, we're looking at the original data and the annotated or the segmented results that you have. Again, we feed this into a particular model and we get a trained model, which now we can input new EM data. And typically what this creates for you is a confidence map. And the reason why it's called a confidence map is that each of these pixels, the intensity of it correlates to how confident the software is that this is your object of interest. So if it's really confident, that intensity is going to be really high. If it's less confidence, that intensity is going to be really low. So that's kind of one of the reasons why the terminology confidence map is used here. So some examples of what that could actually, of deep learning could look like in microscopy. So one example here I'm going to show you is denoising. So essentially the image pair that we input into the model is a low signal to noise image and the ground truth, which is this high signal to noise image. We create a model and then we apply it to new data, and this is the restored output of this model. And in this particular case, we're using a 3D ARCAN data set. This paper has now been published in Nature Methods, so that bioRxiv reference is a little bit outdated. Another application that we can utilize deep learning for is commonly referred to as virtual painting. And in this particular case, what the model does is it takes in a bright field like image and essentially predicts where the location of certain components of the cells are. So here we're looking at the nucleus, the actin, and the microtubule. So we're going from a bright field and predicting where these three components are for each individual cells that is located here in the image. So it's kind of like that conversion between bright field and fluorescent like image with all of these three components. Another application for deep learning is automatic segmentation and EM images. In this particular case, what we're segmenting are neurons in this EM data set. And once the confidence map is created, we can utilize it to create these morphological meshes of what the neuron surface looks like, and from there also create those center lines to allow you to look at complex networks a little bit better, and with the morphological surfaces kind of allow you to see how the neuron dendrites kind of interact face kind of like directly and things like that. So this allows you to kind of go from EM to these meshes and result automatically. So some key advantage that I hope that you've seen with the examples I provided here is the ability to increase cell viability. And this one is really well highlighted in that denoising method, denoising model, because you can take a low signal to noise image that allows you to really preserve your cell viability over a long period of time, but transform it and create a restored image that really allows you to capture the complexity and that crisp image that you may need to answer some of your biological questions and to do your further analysis. Another key advantage is automating difficult tasks and time-consuming tasks. This is really highlighted by that EM model, where now lots of labs kind of go in and manually draw these EM and trace their cells. Now we can apply a deep learning model to really do this automatically. It allows you to kind of enormously increase your output because you no longer need to kind of sit there and do this manually. You can kind of apply the model overnight, over the weekend, and come back to results really quickly. And then the last key advantage I want to highlight is expanding the utility of your microscope. And this is really highlighted by the virtual painting model. So taking a bright field image, again, that gives you a scope that might be a microscope that might be easier for you to access, or you really need that type of system to keep your cells viable and really take that image and transform it and create and predict something that allows you to, again, really delve into the complexity and really allows you to answer some of those research questions that you're thinking of without sacrificing some of the cell viability and some of the microscopes that you may have at hand that are readily available to you. So some key summaries of AI and AI in microscopy overall. You provide examples instead of the rules. You provide examples of what the cells look like, what a good image looks like, what the segmentation results could look like, instead of the rules that govern those key activities and those key detections. It allows you to add complexity and iteration into your analysis that really allows you to expand beyond some discrete measurements and really allows you to tackle some of the more challenging analysis and challenging images that you may have created. and it can be utilized both for image enhancement and image analysis. You can also even combine these two components to really have a full workflow that is AI based. Or you can also combine it with other techniques that are kind of already optimized for your lab and your images, and really take advantage of AI to help you along the way. So with that, I just wanted to mention that if you've been really curious about any of the applications I showcased today, I do highly recommend that you go and request your Avia trial. It's free, and the applications and the videos I showed you today was created in Avia. So we do have the tools in terms of machine learning and deep learning that you saw today. Just a brief highlight because I missed some things in terms of the artificial intelligence capability in Avia. So we talk about machine learning, and we talked a lot about the pixel classifier, but Avia also has object classification that's really driven by the machine learning deep, random forest algorithm. Deep learning here, the models that I showed you, in Avia, we have already created these models, so they're pre-trained, so you can really immediately use them for your applications. So we have that image restoration denoising that I spoke of, the image segmentation, and the virtual staining. One that I didn't get to cover is the image resolution model, which allows you to essentially go from a confocal image to a higher resolution image. So again, really allowing you to perhaps improve your cell viability or really take advantage of the utility of the microscopes that you have at hand while still obtaining the information that you need for further work. So with that, I want to say thank you very much for attending. I hope that was very useful and informative. And now I'm happy to take any questions that you have. Again, thank you very much.
And thank you, Dr. Tran, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of the screen, and we'll answer as many as we have time for. So let's get started. It looks like we already have some great questions coming in from our audience. Dr. Tran, what are the computer requirements for using AI tools? Oops, we cannot hear you. You're going to have to unmute.
Sorry, can you hear me now?
Yep, perfect. Thank you.
Excellent. So one of the key components in terms of computers that you need is a good graphics card, specifically because it's going to utilize a lot of the GPU to do the processing and the learning. Now, the machine learning piece requires a little bit less, while the deep learning requires a lot more capabilities. There are also some systems that are set up to take in multiple graphics cards. So again, that ability is going to kind of speed things up and make things a little bit easier. From kind of a consumer perspective, if you want to get started, a really nice place to start is kind of some of the requirements for gaming laptops, essentially. That's kind of where I started using a gaming laptop, specifically because they're really put together and organized so that it's got a very, very nice graphics card on there, because computer games require quite a bit of that processing. So that's a really good place to start. If you visit our Avia webpage, we also have a section that has system requirements and lists out some specific key components you need, including which graphics card we recommend.
Thank you so much. And Dr. Tran, when does someone use a machine learning versus deep learning?
So that's a really great question, and that varies quite a bit depending on what you're doing. So some of the advantage and disadvantage here is machine learning is a lot easier to kind of get started. So as we kind of saw in the training, all you really need to do is paint examples. So you can essentially use any of the existing image that you have to paint examples. With deep learning, if there are not pre-trained models that work out for you, it's likely that you'll need to create additional data to train and augment that model or to build your own model completely from scratch. So it is going to require you to take a little bit more time at the beginning to generate the model that you need. My recommendation is that if you kind of want to get started right away, I would say try the pixel classifier and the object classifier using machine learning, because again, that barrier to entry is significantly lower. And if that doesn't work, then we kind of move on to deep learning. That's probably the best strategy to begin with.
Thank you so much, Dr. Tran. Now, how about images? How many images are required for the training?
So again, there's quite a bit of variation here, and it really depends on what it is you're looking for and how different they are. So from the machine learning and deep learning perspective, the clearer the distinction between what you're looking for in the background or cell A versus cell B, the less examples that you will need. For the machine learning, again, I'm talking about that picture, that video where we're painting and training. You can see that most of the time the machine learning doesn't require as much of the data as we need. So it's just providing select regions of what the objects look like. For the deep learning, it depends on what you're doing and again, what it is that you're looking for. The denoising model that you saw was created by 10 image pairs. So basically we fed pairs of low signal to noise and high signal to noise, and there are 10 of those sets. The EM segmentation gets a lot more complex and takes a lot more data. So it varies quite a bit. And I highly suggest, again, kind of as a starting point, use that machine learning piece to get started. And then if you're looking to move to deep learning, take a look around to see if anybody's already created a model that is what it is that you're looking for. And what you're trying to find really here is to match the images that you've already created to what they're using to train the model. And if it's a really good match, it's likely that you can use that pre-trained model so you don't have to train your own.
Thank you so much, Dr. Tran. And again, I want to thank our audience for these great questions coming in. Our next audience member asks, does a random forest algorithm only look at a single pixel information or also an example like neighborhood or whole object size?
So the answer here is it looks at all of that. So there are what you create, how the features is created is through a set of processing algorithms that take a look at pixel information, how 2 pixels relate to each other, how 3 pixels relate to each other, and it builds and builds that layer. So it looks at basically everything like that. For the object classifier, it looks specifically at your meshes, so it takes in the whole object. But the pixel classifier also builds that hierarchy and generates not only pixel specific data, but also kind of neighborhood and city-like features. So it's a wide range. Whether it uses all that information to build your random forest is slightly different. It's going to use whatever is best to classify your data. That could be just, how 2 pixels relate to each other, or that could be how the entire cell relates to the next cell. So it varies quite a bit in what gets pulled out eventually, but it does take a look at everything.
Thank you so much. Now, Dr. Tran, what features do you use for random forests?
Now this is kind of a broad selection, and I have to be honest, I don't know which one I use at the top of my head, purely from the perspective that with Avia, which is the software I'm currently using to do the processing and what I've used to showcase the examples here, we do have a set of default features that we use. And this is for cells versus kind of straight lined objects and things like that. So there's already kind of pre-programmed sets that we use, and I don't know what those specific features at the top of my head looks like. I can email that out later, but there is a wide range of features that you can use. A lot of it, if you're using a tool that's common, that's already kind of pre-built for you in terms of machine learning, those features tend to be already programmed in for you. If you really kind of want to go in and tweak which features you use, they usually have the ability to do that. But most of the time, people do tend to set a certain number of defaults for you so that you can at least get started with the process. Again, I can go into more details in an email later, but I don't know that off the top of my head.
Of course, thank you so much. Our next question, can you briefly explain what is the advantage of RCAN compared to UNet?
So this is going to be a slightly it's going to be a complex answer. And again, probably something we want to cover a little bit more in my email, just because I can send you articles for reference and things like that. But overall, the UNet basically kind of has a UNet structure, and it's really good, and it's been widely used for segmentation. Segmentation, kind of like the neuron segmentation that we have, that's been kind of the UNet structure, and that's the one that's really widely used in that application. RCAN tends to work better when we're talking about the denoising application. It's very popular there. So the structure is different, but it's really from a user perspective, it's really about which one works best for the application. And that's kind of been generally how it's been divided. Of course, the UNet structure also can be used for kind of the denoising application. It's used for both. It's one of the first kind of architectures that was widely used. So it's used for everything. But in Avia's case, we really have tailored it to the segmentation portion versus the RCAN that is for denoising. But again, there's a lot of details here that I can provide via email.
Thank you so much, Dr. Tran. We have time for one more question. You showed a confidence map, which looks a lot like a confidence interval, that a given pixel is statistically correlated with each component extracted. Does the underlying algorithm utilize principal component analysis?
So in Avia's particular case, we do not do that. We do not use a PCA. However, I've seen lots of other works that takes an additional step forward and utilizes that particular, the PCA principle to do further processing. So no, in this case, it does not.
Dr. Tran, thank you very much for your presentation. Would you like to provide any closing remarks for our audience before we go?
Of course. I just, again, want to kind of reiterate a big thank you for everybody who's attended. I hope that this was really informative for you. And, you know, there's a lot left here to cover. This is just, I feel like, the bare minimum to kind of get us started. So I really highly recommend for anybody who's doing some challenging data analysis and challenging visualization to try out some of these tools and to try out AI, because I've seen a lot of amazing work come out of that. There's also a ton of literature out there, so this is just the flavor. If you want to, you know, start to explore it a little bit more, there's lots left to do. And especially on the deep learning side, there's a lot of capabilities that are still being explored. So I think for the next 5 to 10 years, we'll see some really exciting work come out in terms of AI in application to the microscopy field. So be on the lookout. It's going to be really exciting. And again, just thank you for attending, and thank you, LabRoots, for kind of putting all of this together.
Well, thank you, Dr. Tran, again for your time today and for your important research. And I also want to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted on the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay safe. Bye-bye.